Bacteria are building with iron. Right now, in cold water, no heat, no pressure, no machines. To do what they do, we need furnaces hot enough to melt rock. We need coal. We need infrastructure that costs hundreds of millions of dollars and produces carbon dioxide at every step. They have been doing it longer than animals have existed on Earth. We have known about it since 1836, and we still do not use it. Why? Because what these bacteria do, we still cannot replicate in a factory, not because it does not work, because we do not know how to scale it, and what they are doing sounds simple until you realize that when iron forms naturally in water, it turns into useless sludge. When bacteria do it, they build structure. The story of how we found this and failed to use it starts in 1836. In 1836, a German naturalist named Christian Gottfried Ehrenberg peered through a microscope at water samples from iron-rich springs. He was looking for fossil microorganisms, tiny creatures he thought might have been preserved in the sediment. What he found instead did not fit any category he had. A microscopic organism, alive, surrounded by rust, not living in rust, not stained by rust, producing it continuously, as if rust were the point. He assumed it was a fossil, named it Gallionella ferruginea, published his findings and moved on. The scientific community filed it under curiosity. We were building railroads, we had blast furnaces. Why would anyone look twice at a bacterium making rust in a cold spring? For most of the next century, that's essentially where it stayed. Then, in the 1950s, something shifted, not in the laboratory, but in the ground. It reframed the entire iron industry without anyone quite realizing it at the time. Geologists studying the world's great iron ore deposits, the banded iron formations, started asking an uncomfortable question. These formations record a period roughly 2.5 to 1.8 billion years ago, when the oceans were saturated with dissolved iron. And then, over millions of years, that iron precipitated out as solid mineral layers. The standard story had been purely chemical, no biology required, but the chemistry didn't quite add up. The isotopic signatures, the mineral phases, the rates, they kept pointing toward biology as at least a contributor. Whether the iron ore we mine today is partly the exhaust of microbial metabolism from 2 billion years ago was a question that forced a reckoning. If iron-oxidizing bacteria built a significant fraction of Earth's iron ore deposits, they were not a curiosity, they were a geological force. Think about what that means for a moment. The steel in a bridge, a car, a ship started as ore dug from layered rock that formed when ancient oceans were thick with invisible dissolved iron. That iron didn't precipitate on its own. Something was living in those oceans, stripping electrons from iron ions, using the energy to survive and leaving behind solid mineral as waste. The rock record we mine today may be, in part, the accumulated biological exhaust of organisms that lived and died before there were animals on Earth. We smelted it. We built civilization with it, and we had no idea where it came from. By the 1970s, the tools had improved enough to start seeing what these organisms actually produced. Scanning electron microscopy revealed the Gallionella stalk in detail, including the helix, the nanoscale dimensions, and the mineral composition. And researchers had begun noticing something stranger inside certain bacteria. Some of them moved consistently in the direction of magnetic north. An Italian microbiologist named Salvatore Bellini had first observed this in 1963, watching bog sediments under his microscope in Pavia. He published in Italian in a regional journal. Almost no one read it. Then in 1975, a graduate student at the University of Massachusetts named Richard Blakemore, working at the Woods Hole Oceanographic Institution, made the same observation independently. Bacteria moved in one direction and reversed when he brought a magnet close. He followed the observation to its source and found what no one had expected. Chains of tiny perfect crystals of magnetite inside living cells. Biological magnets assembled at the nanoscale by a single-celled organism. He published in Science, and this time the right people read it. That discovery landed differently than Ehrenberg's had. By 1975, materials science knew exactly what magnetite nanoparticles were, and it knew how hard they were to make with consistent properties. The idea that a bacterium was producing them reliably, continuously at room temperature, in water, was not filed under curiosity, it was filed under how. That question, how biology does this and whether we can use it, has been driving the field ever since. We spent over a century dismissing the slow, small, ambient thing in favour of the hot, powerful industrial thing. It turns out to have been one of the more expensive blind spots in the history of materials science. So what exactly have we been ignoring? Let's make that contrast concrete, because the scale of the difference is the whole point. You dig ore out of the ground, you crush it, you smelt it above 1,500 degrees Celsius. You add carbon, remove impurities, pour molten metal into moulds, let it cool, machine it, treat it. From ore to steel beam, enormous energy. Carbon dioxide at every step. Industrial equipment that most of the world can't afford or access. Expensive, hot, dirty and centralized. Now look at what's happening in a cold stream somewhere right now. Microscopic bacteria taking dissolved iron, invisible, scattered ion by ion through the water and assembling it into solid mineral structures. At room temperature, in water, powered entirely by chemistry that costs nothing and stops for nothing. Not metaphorically, literally. It has been happening longer than animals have existed on Earth. To understand what these organisms are actually doing, you need to understand one thing first. Iron is invisible in water. Not scarce, not absent, invisible. It dissolves into water as charged particles, ions, and disappears. A glass of water can be saturated with iron and look completely clear. You would drink it and notice nothing except maybe a faint metallic taste. You have probably seen what happens next without knowing it. Fill a glass from a well, leave it on the counter, and sometimes it turns faintly orange within an hour. That is the dissolved iron reacting with oxygen dropping out of solution as a solid. The orange is iron oxide, rust, assembled from nothing visible in water at room temperature with no help from anyone. That spontaneous process is called oxidation. And here is what is important. When it happens randomly, you get a mess, an amorphous slurry of iron oxide particles with no particular shape, no particular structure, no useful properties beyond vaguely orange. When bacteria do it, you get something completely different. The same chemistry, dissolved iron becoming solid iron oxide, but organized. Structured at the nanoscale, with geometry that encodes function, with properties that industry spends billions trying to replicate and still cannot match. That is the whole story. Everything else is detail. Iron in water exists as invisible ferrous ions, the dissolved form. Bacteria oxidize them to ferric ions, using the energy released to power their metabolism, and leaving behind structured iron oxide minerals instead of a random orange mess. Spontaneous oxidation produces disorder. Biological oxidation produces architecture. That is the distinction that matters. The formal term for what these organisms do is biomineralization. Living things producing minerals as part of their biology. You already live with this. Your bones are calcium phosphate deposited by your own cells into a precise matrix that is simultaneously rigid and capable of ongoing repair. Seashells are calcium carbonate, assembled by mollusks into layered structures with fracture resistance that materials scientists have spent decades trying to replicate. Diatoms, single-celled algae, build silica shells so geometrically exact they are used to calibrate electron microscopes. Iron biomineralization is the same principle, but the organisms doing it are bacteria. Single-celled, microscopic, without a nucleus. And yet the structures they leave behind are, in some environments, geologically significant. Visible. Old enough to be preserved in the rock record. The mechanism is stranger than it sounds. Meet Gallionella ferruginea, the one Ehrenberg found. A bacterium. Lives in iron-rich groundwater and freshwater springs, cold, dark and oxygen limited in a very specific way. Gallionella does not want zero oxygen, it wants precisely a little. It lives at the interface between oxygen-rich and oxygen-poor water, where dissolved ferrous iron flows up from anaerobic depths and oxygen diffuses down from above. That narrow chemical boundary is its entire world. It builds a twisted helical stalk made of iron oxide, not as a side effect, not accidentally. The stalk is the exhaust. Gallionella strips an electron from ferrous iron to produce ferric iron and uses the energy released to power its metabolism. The iron oxide minerals that form are what is left over, except the exhaust is organized, structured, helical. Why helical? The organism rotates as it moves through water and the stalk deposits continuously behind it, encoding that rotation into the mineral structure itself. Under a scanning electron microscope, it looks like a metallic corkscrew. Iron oxyhydroxide minerals assembled atom by atom at ambient temperature in water by a living thing. Each stalk is roughly a micron wide, about 100 times thinner than a human hair. There is a detail that takes a moment to absorb. The stalk is not just shaped by the organism's rotation, it is a physical record of the organism's journey. Every twist in the helix corresponds to a movement the bacterium made. The mineral structure is essentially a diary, written in iron of where the organism has been. When researchers image a Gallionella stalk, they are reading the travel history of a single bacterium, preserved in solid mineral at nanoscale resolution. No human technology produces anything like this accidentally. Here is the part that stops materials scientists, that helix is not decorative. The twisted geometry is a direct consequence of how it was made, the organism's rotation encoded into the mineral structure as it deposited. No mould, no template, no external force shaping it. And the stalk it produces is an inorganic-organic hybrid with a carbon core and iron oxide surface. A structure with porosity and reactive surface area that researchers are still characterizing. You cannot get that architecture by precipitation alone, and you cannot get it with a furnace. This is what biological oxidation produces, not the amorphous orange slurry you get when iron oxidizes in a glass of well water. Architecture, at the nanoscale, assembled continuously in cold water for free. And then there's a completely different category, magnetotactic bacteria. This is where Blakemore comes back in. The organisms he observed, species like Magnetospirillum magneticum, don't use iron for energy, they use it for navigation. Inside their cells, they build tiny crystals of magnetite, iron 3 oxide, organized in precise chains within membrane-bound compartments called magnetosomes. Each crystal is typically 35 to 120 nanometers across, roughly 500 times smaller than a red blood cell. They are single-domain magnets permanently magnetized. The chain acts as a compass needle, letting the bacterium orient with Earth's magnetic field to navigate oxygen gradients in the water column. This sounds almost too elegant to be real. A bacterium that builds its own compass, crystal by crystal, inside its own body, calibrated to the planet's magnetic field. But it gets stranger. The crystals are not just magnetic, they are the perfect size to act as a stable magnet, and that size is extraordinarily precise. Too small, and the crystal will not hold a stable magnetic orientation. Too large, and it breaks into multiple internal domains that cancel each other out. Every single crystal lands in the exact window where a single stable domain can form. Not because the cell is measuring them, but because the biosynthetic machinery that builds them is tuned through evolution to produce crystals in exactly that range. The organism has solved, without knowing it solved anything, a materials engineering problem that took physicists decades to fully characterize. What makes this extraordinary from a manufacturing standpoint is the consistency. Magnetite nanoparticles in that size range with uniform crystal orientation, produced at room temperature from dissolved iron, that is a level of nanoscale control that synthetic chemistry still struggles to match at equivalent cost. Achieving that combination of size, consistency, crystal quality and magnetic uniformity reliably and cheaply remains an active research problem. The bacteria have had it solved for at least 500 million years. This should have changed manufacturing, it didn't, and this is where the story breaks. We opened on cold water, that detail wasn't atmospheric, it's the point. The iron smelting that built civilization requires temperatures that would incinerate any organic material. These organisms do related chemistry in water cold enough to drink. No pressure, no exotic inputs, iron, oxygen, water, the three most common things in the near-surface environment of this planet. And the process never stops, no batches, no downtime. The Gallionella stalk keeps growing, the magnetosome chain keeps assembling. In the right environment, you don't run a process, the process runs itself. The output isn't random. The helical architecture of the Gallionella stalk is encoded in its growth process, geometry as a product of biology. The magnetosome crystals have magnetic properties that emerge from biological control over nucleation at the nanoscale. This is organized output from organized biology, bottom-up manufacturing running continuously in the cold, powered by chemistry that asks nothing in return. So why aren't we using it? This isn't about replacing steel. Steel is cheap, scaled, and works. What these bacteria offer is something different, the ability to build structures at the nanoscale in specific environments in ways industry still can't match. The question is whether we've figured out how to use that. Let's start with what we are actually using, because there is one real deployed application, and it matters more than it sounds. Drinking water. Across large parts of Europe, Asia, and sub-Saharan Africa, groundwater has high dissolved iron content, enough to stain laundry, corrode pipes, and make water taste like metal. The biological fix has been quietly running in the Netherlands since the 1950s. Slow sand filters are colonized by Gallionella and related bacteria, removing dissolved iron continuously without chemical inputs and at lower operating cost than chemical treatment. Deployed municipal infrastructure. Real cities. It works. Almost nobody outside the field knows it exists. That's the proven end. Now here's where it starts to look like we might use this more broadly, and then it breaks down. In nanomaterials, research groups are cultivating magnetotactic bacteria to produce magnetite nanoparticles for medical imaging and drug delivery. The biological particles have properties synthetic methods struggle to match, including narrow size distribution, high crystallinity, consistent magnetic behavior, and natural biocompatibility. The biology is real. The manufacturing cost isn't yet competitive, but for high-value medical applications, the economics are closer than they look and narrowing. In contaminated site remediation, Geobacter sulfur reducens and related bacteria can immobilize uranium in groundwater by coupling iron reduction to precipitation of uranium as an insoluble mineral. Field trials at Department of Energy sites in Rifle, Colorado, and Oak Ridge, Tennessee, have shown that stimulating native Geobacter populations can reduce dissolved uranium significantly within weeks. It works slowly, requires careful management, and gets used primarily where traditional remediation is impractical or impossibly expensive. One proven application at scale, two more where the biology works but the deployment doesn't, and a much larger set of possibilities that researchers have been tantalizingly close to for decades. So the question isn't whether this biology is useful, it clearly is. The question is why the gap between this works in nature and this works in a factory has stayed so stubbornly, frustratingly wide. None of that matters if you can't actually build with it at scale. Why haven't we cracked this? The short answer is that biology doesn't scale the way we assume it does. The longer answer is the one that actually matters. When we see what a single cell can do, the precision, the self-organization, the nanoscale control, the instinct is immediate. Just use more cells, more volume, more time. If one bacterium builds a perfect magnetosome crystal, a trillion bacteria in a tank should produce a meaningful quantity of them. The arithmetic seems to hold. It doesn't. Imagine you found a master watchmaker who produces extraordinary work. You want to scale up, so you hire 1,000 watchmakers and put them in a factory. You discover immediately that the factory is not 1,000 times better than the workshop. Coordination problems, quality variation, shared resources, the factory becomes a fundamentally different system. You can't simply multiply. Biological scaling is that problem, but harder. You are not hiring independent craftspeople. You are growing a population of organisms that interact chemically, compete for resources, and change behavior based on signals from neighbours. The cell that produces perfect magnetosome crystals in isolation may produce something different entirely when surrounded by a billion copies of itself in a tank. If this were easy to scale, we wouldn't be using furnaces at all. The chemical gradients that guide crystal nucleation in a single cell are maintained by the cell's own machinery, membrane transporters, protein scaffolds, ion channels tuned by evolution to create exactly the right local environment. In a tank, those gradients do not exist the same way. You have bulk chemistry, statistical averages. The exquisite local control that makes single cell output extraordinary becomes noise at population scale. Here is the part that surprises most people. The bacteria in that tank are not failing, they are thriving, growing fast, metabolizing iron, doing exactly what they evolved to do. The problem is that what they evolved to do and what you need them to do are subtly but consequentially different things, and at scale, subtle differences compound into unusable output. Speed, control, environment, economics. Every specific obstacle you hit when trying to use these bacteria industrially traces back to the same root problem. Speed biology is slow, and running more of it in parallel hits the scaling wall rather than returning linear gains. Control, the magnetosome uniformity that makes magnetotactic bacteria interesting, is a property of cellular machinery, not just genetics. Modifying the genes does not guarantee the output if the cellular environment at scale does not support it. Environment, Gallionella needs specific iron concentrations, a specific pH range, low but non-zero oxygen. Maintaining those conditions uniformly through a large volume of water is a completely different engineering challenge from controlling a blast furnace. Economics, steel is cheap not because it is easy to make, but because the infrastructure has been amortized across a century of production at scales that crush cost per ton. For biology to compete, it has to produce something industry genuinely cannot, or operate where industrial infrastructure simply does not reach. Here is what makes this particularly frustrating. In the early 2000s, multiple research groups, including teams at Université Paris Sud and collaborators in Japan, set up exactly the kind of bioreactor you would imagine. Large tanks, magnetotactic bacteria, carefully controlled chemistry. The bacteria grew, the crystals formed, and the output was inconsistent in ways that took years to diagnose. Crystal size varied. Magnetic properties drifted batch to batch. Some of those problems are still not fully solved. So what does the frontier actually look like? Because it's not empty. The questions being asked now are genuinely different from the ones being asked 20 years ago. Researchers at several European and Japanese institutes are working on magnetically guided biofilm reactors. Using external magnetic fields to organize magnetotactic bacteria into structured spatial arrangements, imposing order on the population using the bacteria's own navigational instinct. If you can control where they are, you can influence the local chemistry around them. Early work, promising at bench scale. At MIT and collaborating labs, CRISPR tools are being used to modify the magnetosome biosynthesis pathway, not to change what the crystals are made of, but to make the process faster and less sensitive to environmental variation. The pathway involves dozens of proteins interacting in a precise sequence. Modifying one often disrupts others in ways that take years to trace. Progress is real, the pace is slow, none of this is a breakthrough, all of it is movement. And the stakes of getting it right are worth being clear about. If magnetotactic bacteria can be coaxed into reliable continuous production, the medical imaging industry gets a biocompatible nanoparticle it currently can't make consistently. If Gallionella can be deployed at scale for water treatment, hundreds of millions of people with high iron groundwater get infrastructure that runs without chemical inputs indefinitely. If iron-reducing bacteria can be reliably directed in contaminated sites, the cost of cleaning up the most intractable industrial legacies drops dramatically. None of these is a moonshot. All of them are plausible within the gap we're trying to close. What's changed underneath all of it is the tools. Sequencing a bacterial genome used to take years and cost millions. Now it takes days and costs hundreds. Imaging the interior of a living cell at nanometer resolution. well enough to watch a magnetosome crystal nucleate in real time, is routine in well-funded labs. We can read the genome of an entire iron cycling microbial community from a thimble of water in a week. The gap isn't knowledge anymore, it's engineering. And iron biomineralization isn't the first time we've been stuck in exactly this position. Spider silk is stronger than steel by weight and tougher than Kevlar, produced at room temperature from water and protein. We've understood the mechanism for decades. We still cannot manufacture it at scale, not because we don't know the chemistry, but because the biological spinning process that produces the fiber's properties is so tightly coupled to the organism doing it that replicating the output without replicating the biology has proven extremely difficult. The pattern is the same every time. Understanding the mechanism doesn't translate into using it, not from ignorance, but from the distance between what evolution optimized for survival and what engineering needs for repeatable industrial production. Evolution doesn't care about throughput or batch consistency. It optimizes for one thing and everything else is a byproduct. Iron biomineralization sits squarely in that territory. The biology is extraordinary. The mechanisms are understood in remarkable detail. What separates understanding from deployment is real, wide, and full of engineering problems without obvious solutions. That gap is closing, with synthetic biology tools sharpening, genomics making biosynthesis pathways legible, microfluidics enabling study at controlled intermediate scales. The pace is slower than the optimists predict and faster than the skeptics allow. Anyone who tells you the answer is just around the corner is selling something. Anyone who says it's hopeless isn't watching the tools. There are bacteria at the bottom of cold streams right now, building iron mineral structures more precisely than most of our nanofabrication processes can manage. Ehrenberg saw them in 1836 and filed them under curiosity. Bellini watched their magnetic cousins swim north in 1963 and published it where no one would read it. Blakemore found them again in 1975 and the field finally asked how. We've spent nearly 2 centuries moving from curiosity to how, and we're still not done. We've deployed one application at scale, demonstrated several others, and built tools sophisticated enough to finally start understanding, not just observing, the distance between what biology can do and what we can harness. The question was never whether this works. It works. It's been working in the dark in cold water, without stopping, since before there were animals on land. We understand what they're doing, we just don't know how to do it ourselves. And until we do, this might be one of the most advanced manufacturing systems on Earth, running quietly in the dark without us.